Lecture Series. So we are very thankful for your attendance today. And for everyone that's already in the Zoom, we will wait for a few minutes for the admission of the other participants, and we will start our program officially at 9:05 a.m. Good morning, everyone. We would like to welcome all our participants for today's lecture. The first lecture, we have Dr. Tami Iwasa Aray to discuss about whale lice. And today we will have another lecturer who will discuss about plankton ecology. So for today, we will be starting with the opening remarks or the welcome remarks by Dr. Mark Louis Lopez, the current administrator of Philippine Planktology.
Hello, can you hear me okay? Hello, everyone. So I'll go straight to the point. I'd like to welcome you to the second leg of our lecture series, which is the fourth Philippine Planktology Lecture Series. This is in collaboration with the Institute of Marine Fisheries and Oceanography at the University of the Philippines in Visayas. And we're so grateful for this partnership. They've been hosting this event for the past 4 years. This is also an activity in collaboration with the Philippine Plankton Society and the Plankton Niche Laboratory based at University of Visayas. We're also live streaming via Facebook. So if you have questions, let's wait for further instructions from Darlene as we proceed with the program. So I hope you enjoyed this talk because we have a very great speaker from Vancouver, Canada. Yeah, so welcome everyone and hope you learned something.
Thank you, Sir Louis. Before we start with the lecture of our guest speaker today, we would like to remind everyone of a few house rules in order for us to have a very orderly lecture for today. So first, Participants are requested to turn off their cameras and keep their microphones on mute when not speaking. And then please also hold your questions until the end of the presentation because questions will be entertained during the open forum. And then to ask a question during the open forum, please kindly raise your hand and wait for the moderator to call you in. And then you may also type in your questions in the Q&A box, and please do not spam the chat box. Lastly, please accomplish the evaluation form on the link provided in the chat box to claim your e-certificate for today's lecture. So we have a lot of students, both undergraduate and graduate students in our participants today, and of course, other members of both private and government institutions. And I'm sure everyone is very excited to listen to the discussion today. So without further ado, let me call in Dr. Christy Acabado, the current director of the Institute of Marine Fisheries and Oceanology, and also the vice president of the Plankton Society of the Philippines, to formally introduce our speaker for today's lecture. Ma'am.
Good morning. Good morning, everyone. I am happy to welcome a Filipino expert on zooplankton ecology today. He is a postdoctoral researcher in the Pelagic Ecosystems Laboratory at the University of British Columbia. He received his PhD in oceanography from the University of British Columbia in 2024 and MS in Marine Science from the University of the Philippines, Diliman in 2017. Dr. Patrick is a biological oceanographer focusing on zooplankton ecology, and he is interested in understanding the oceanographic and ecological drivers of zooplankton distributions and how the emergent spatial-temporal patterns translate into ecosystem functions and services. His research involves ecosystem modeling and the synthesis of observations of water properties, plankton communities, and organismal traits. Everyone, please help me welcome our speaker today, Dr. Patrick Pata.
Hi, everyone. Thank you to Christy and Louis for this invitation. And yeah, I'm very excited to give you this talk. I will first gonna try to share my screen. Yep. Does this look okay?
Looks
Perfect.
good, Patrick.
Okay, great. So good morning. Thank you for attending this talk. This is going to be about bridging species level and community level processes using trait-based approaches in zooplankton ecology. Okay, so the title sounds quite dense. So let me start with this photo to give you an idea of what this lecture is going to be about. So what you are seeing now are two zooplankton individuals. On the left in the foreground is an amphipod. You can see that it has its claws out. You can see its eyes on the top, and it's seemingly staring at what appears to be a copepod on the right side. So let's try to interpret this photo and imagine what this that this was taken right before the moment when this carnivorous amphipod attempted to eat that copepod. So whether or not the amphipod successfully feeds on the copepod is dependent on the capabilities of the amphipod as a predator, and it is also dependent on the capabilities of the copepod to evade predation. So if we dissect the moment of interaction between these two zooplankton individuals, we can break down the feeding process into the steps from the predator encountering its prey to pursuing it, to capturing it, handling the prey, ingesting it, and eventually digesting and assimilating the prey into its body. So for each of these steps of the feeding process, we can think of the various biological and environmental factors that affect each of the steps. So for example, the event of encountering the prey is dependent on what are the perceptive capabilities of the predator. It's also dependent on the turbulence of the water, prey concentration, and the prey's escape response. So when we break down the processes into their core parts and figure out how they interact, we get to understand the underlying mechanisms of ecology. And even for very what seemingly simple processes like feeding between a predator and its prey. So approaching ecological questions through the point of view of individuals highlights the idea that ecological interactions occur at the level of the individual. So what this scenario between the amphipod and the copepod illustrates is that at the individual level, where an organism responds to its environment and to other organisms, and that is all dependent on the traits of an individual. When we aggregate the interactions across the individuals in a population and across populations of different species, these interactions would scale up from the individual level to processes at the community and ecosystem level. So this lecture will be circling around the general themes of traits and bridging the levels of biological organization. And I will give various examples of how a trait-based approach guides how we can look at ecological processes and phenomena through the perspective of individuals. And of course, this applies to any biological entity or system, but it is in my very biased opinion that trait-based approaches are especially useful when we study zooplankton. Okay, so first, what is a trait to begin with? A trait is simply a measurable characteristic, whether morphological, phenological, physiological, behavioral, or cultural, of an individual organism that is measured at either the individual level or other relevant levels of organization. So examples of traits for a copepod include, say, body length, respiration rate, reproduction mode, the size of its eggs, clearance rate, which is like how much water it processes, its carbon weight, carbon content, and the nitrogen content. So those are examples of traits. Another common term is a functional trait, and you may have encountered that. And when we say, when you use the term functional, that implies that a trait is explicitly related to a specific ecological function. So through trait-based approaches, so at the perspective of the individual, taxonomic entities, the species that we work with, can be abstracted to the set of traits that make up an organism. So each individual, including you and me, is composed of key elements, and the configuration of how much carbon, nitrogen, phosphorus, and water that is contained in an organism affects its morphology, such as body size, shape, and transparency. Organisms maintain their internal stoichiometry, or the ratio of the different nutrients in their body, by consuming food, which is quantifiable through its feeding rate. And different organisms have different strategies in acquiring nourishment, which affects what trophic group we classify them into. So we either classify them into, say, herbivores or carnivores or omnivores, right? And then organisms metabolize food through respiration and release dissolved and particulate compounds through excretion and egestion. Then, like in the process of assimilating these nutrients, organisms assimilate the elements as they grow through different life stages, and also for reproduction, which would connect these processes that are happening at the level of the individual to the dynamics at the level of populations. So these variables are measurable traits that are unique characteristics for each species. And different species, from copepods to salps to euphausids, to theropods, hydromedusae, and ketognaths, or whatever zooplankton you can think of, would all have different values for these traits. Thus, when working with trait-based approaches, a challenge is that we need to know the trait values for different species in a community. And that could be even more challenging when we are working with very taxonomically diverse communities, such as when we're studying zooplankton. The advantage of doing so, of using trait-based approaches, however, is that the traits and processes at the individual level can be explicitly linked to ecosystem functions. So here you have the processes of respiration, consumption, predator avoidance, and growth. And these are linked to like larger community level and ecosystem level functioning, such as ecosystem level respiration, primary production, total biomass, carbon sedimentation, like zooplankton biomass. So fortunately, in zooplankton ecology, there are a lot of fundamental work and data that has been already available in identifying which functional traits of an organism affect the individual level processes. So here on the upper part, we list all the various traits of zooplankton. And then this framework also lists the different traits the different ways that these traits affect ecosystem level functions. For example, how the traits on growth and reproduction and consumption affect zooplankton biomass or biogeochemical cycles. Thus, we have the tools that we need to connect organismal traits and functions to community level and ecosystem level functions and services. And by ecosystem services, I mean the different ecosystem functions that affect human communities. So when we bridge the individual level to the community level, we are trying to understand how the traits are affecting the different ecological mechanisms. So you may not be familiar with trait-based approaches, but it is very likely that you have encountered like a trait-based approach or perspective before, even if the studies did not explicitly specify that they are using traits. So I will first give a couple of examples of this framework to give you an idea of what I mean by bridging the different scales of biological organization and scales of understanding the ecosystem using traits. Okay, so one example is like how traits affect dispersal. So you may be familiar with the dispersal of different larval organisms. So here on the left side is a survey of the different larval zooplankton or meroplankton. So when we look at dispersal and traits, we look at the traits of developmental duration and swimming speed. And we know that the pelagic larval duration, or the time it takes for between reproduction and settlement, affects the dispersal distance of organisms, and that would eventually affect habitat connectivity between different populations. And when we look at the various connectivities of different populations and different meta communities, that's how we get to that affects our estimates of biodiversity. So another example is, for example, we can look at colony shape in coral. So like colony shape is a trait. Different corals have different shapes. For example, there are encrusting corals, there are tabular corals, there are branching corals. And the trait of colony shape would affect coral cover and also habitat complexity. And so these are community level properties, and coral cover and habitat complexity affect ecosystem functions such as fish biomass or wave attenuation. And so like another example you might be familiar with is with phytoplankton. So with phytoplankton, we can look at their taxonomy, but we can also just like simply look at, say, cell size, nutrient requirements, and growth rates of different phytoplankton cells. And all those would affect our primary production, which eventually would scale up to other ecosystem services such as food availability or carbon uptake. So there are many ways in reframing the research that we encounter, the research we do through a trait-based approach. So now let's focus on zooplankton and first consider why we apply trait-based approaches for zooplankton. We know that zooplankton are important for two general roles in the ocean. One is pelagic food webs by transferring energy from primary producers to higher trophic levels. And second, they are important in the ocean's biogeochemistry and in the biological pump as they aggregate carbon at the surface of the ocean, which is then passively and actively transported to the deep ocean. And they also influence the recycling of nutrients, which would fuel primary production. So most of the animals in the ocean are actually zooplankton, or once in their life cycle, like a zooplankton. Thus, by their overall abundance and biomass alone, they affect the entire ocean ecosystem. And a trait-based approach provides a framework to quantify their ecological importance. So for me, a trait-based approach is a necessary framework because especially for very small organisms such as plankton, we often default in thinking about the aggregate or the aggregate level plankton. For example, we study total biomass or total primary production or even taxonomic community composition. And that is partly influenced by how we typically observe plankton, which is typically as assemblages that we sample using a plankton net. So when we observe and study zooplankton, we often work with a diversity of life forms that have different functional roles across a wide range in body size, from unicellular protists to jellyfishes that you can see with your naked eye. And it takes a lot of work to study zooplankton, from going out to sea to collect them, to doing microscopy, to incubating live organisms and running experiments. So we often end up with, so after all of this work, we often end up with data such as taxonomic counts of abundance and biomass, and maybe the taxonomic community composition. Then we try to interpret what all of that means. And that also, the process of analyzing that takes a lot of effort, especially when you're working with tens and hundreds of zooplankton species, and you're trying to figure out how they're all ecologically significant. So yet it is typical for us zooplankton ecologists to struggle in explicitly linking biomass and taxonomic composition with ecosystem functions, and in quantifying the actual ecological importance of zooplankton. So like, what would you do when you know that, your zooplankton community during the spring is like 20% copepods, 50% euphausids, and then how is that different between a community that's like 30% copepods, 40% euphausids? So we need a way to move from looking at understanding community level processes qualitatively to using quantifiable measures. So I'll give you an example of that for how a trait-based approach complements zooplankton taxonomic analysis by focusing on the traits of individuals. So let's consider the organismal traits of lipid reserves. So you can see the lipid sacs for these two copepods here. So these are the lipid sacs of the copepods. And then let's also consider the behavioral trait of diapause or seasonal vertical migration, which is common in high latitude copepods, wherein they would migrate to deeper waters during winter to hibernate. So we know that these traits are important for the organism's growth and survival, especially during the winter. They affect ecosystem function in terms of the overall energy storage of zooplankton, which would eventually affect fish biomass. So when we map out the distribution of diaposing copepod species, we see that it's strongly associated with the distribution of cod and lipid-rich forage species, which makes these environments good feeding grounds for larger organisms such as whales, and also productive fishing grounds for people. Another ecosystem service to look at is through the ecosystem function of vertical flux. So through moving lipids, which are carbon-rich molecules, from the surface of the ocean to the deeper waters during diapause in winter, that affects the vertical flux of carbon, which also affects the global scale carbon sequestration driven by zooplankton migration. So that is just like one example of connecting traits at the individual level to community level processes. And I'll give a couple more later. But another thing to keep in mind when doing these studies is that applying trait-based approaches requires trait data. And often when we need trait data, we need a lot of trait data. Fortunately, zooplankton ecology has a rich history of recording trait information. So there is actually data available, although previously that has not been easily accessible. So in my research, we aggregated decades of trait data into a global zooplankton trait database. The database currently contains more than 50 traits on zooplankton morphology, biochemical composition, physiology, behavior, life history, for various zooplankton taxonomic groups for more than 3,500 species. So there are varying degrees of data coverage for the traits. So in this figure, in the y-axis, you have a list of the different traits. So it's starting with body length and the trait of myelination, carbon weight, eventually into hibernation at the very bottom. Yeah, and then the x-axis is the number of species with available trait data. So overall, the trait of body length has the most number of trait records, followed by categorical traits, such as, say, vertical distribution, mode of reproduction, like trophic groups or feeding mode, etc. So the different traits have different levels of data availability. This time, looking at the coverage as the number of trait records per species, with each of these like lines representing a species along the zooplankton phylogenetic tree. The longer lines indicate that there are species that are well studied, such as large copepods. You know, you have euphausids are very abundant in polar oceans and in shelves, and then like some of the gelatinous organisms are also very much well studied, like say ctenophores or scyphozoans are quite huge and easy to do experiments with. So the percentages listed in these for each of these groups show that for all zooplankton groups, species with more than 5 trait records are actually very limited. So there's not enough, there's not much trait coverage when you look at each of the individual species. And there are definitely gaps in the coverage of trait data. But with over 57,000 individual level trait records and 14,000 species level trait records, there's a lot of data to work with already. However, expanding the database to include more trait records and understudied species, especially tropical zooplankton species, is a very important next step for the global zooplankton trait database. So I invite you all to explore and use the trait database. The database is currently stored in our lab's GitHub account and versioned through Zenodo, and we're working towards connecting this with database managers. You can freely access it through the link or the QR code provided. So with a big database like this, there are a lot of applications, but now I'm just going to show you just a couple of ways we have applied this in our research. Okay, so one obvious application is in identifying zooplankton functional groups. And you may have read similar stuff that are doing this for other domains of like ecological systems. We applied this to the species of the Northeast Pacific Ocean, including 163 zooplankton species from 7 phyla. So including copepods and then soft-bodied organisms and gelatinous organisms. So we extracted the trait data for all the 163 species from the trait database, and you can see a list of the 8 traits that we consider that are important aspects of individual level processes. So that includes the traits of dry weight, water content, respiration rate, ammonia excretion rate, clearance rate, which is related to feeding rates, and then reproductive strategy, and then feeding strategies. So from that, we use a cluster analysis to identify 10 zooplankton functional groups with similar characteristics. And these groups include species from different taxonomic groups. So I'm gonna try to zoom into this dendrogram. Right. So as an example of identifying functional groups, on the left half are species that are current feeders. Functional group 1 includes small current feeders that include small crustaceans, but also small soft-bodied current feeders, which use a mucus mesh in filter feeding, such as thecosome theropods and appendicularians. Similarly, the larger zooplankton size classes include soft-bodied current feeders, including your salps. There are large crustacean current feeders, such as large geophosids, and also like the large copepods common in higher latitudes. Functional group 4 are partly current feeders, but instead of having a more herbivorous diet, they are more detritivorous or carnivorous. So these are mostly crustaceans such as copepods and also ostracods that a lot of these live in deeper waters. So on the right half of this dendrogram, functional groups 5, 6, and 7 are large, soft-bodied carnivores from different phyla that utilize different feeding strategies. So there's a separate group of soft-bodied cruise feeders. So these are raptorial feeders, including polychaetes and some of the jellyfish that are more raptorial. There are different 2 groups of ambush feeders. There are soft-bodied ambush feeders, which are chaetognats, and then soft-bodied passive ambush feeders, including siphonophores and some of the other hydromedusae. And then finally, on the bottom half are species that instead of doing broadcast spawning, they brood their eggs. So functional group 8 include quite small copepods, especially copepods such as Orthona, which are very ubiquitous in the global ocean. And then functional group 9 are large brooding omnivores, including mysids. And then functional group 10 are other brooding carnivores, such as some of the copepods and amphipods. So it may seem that these functional groups only describe their size and behavioral strategies. But if we compare the groups based on numerical traits, so traits that we can measure, there are many dimensions in which the groups noticeably differ. So each of these subplots represent a trait. So this is a plot for body length and dry weight. And here we're comparing the different functional groups. So you can see how, say, functional groups 1 has like lower body length in comparison to functional group 2, which has a higher body length. So one thing I can highlight is that some pairs of functional groups might be similar in terms of size. For example, the 3 soft-bodied carnivorous functional groups 5, 6, and 7, are more similar in terms of body length and dry weight. But when we look at other rates, such as respiration rate, also basically the amount of oxygen they consume in an hour, and then clearance rate, so their feeding rates, we see that they could be quite different. For example, functional group 7 has much lower respiration rates, especially since it's a passive ambush feeder. So it basically does not move that much. And so it does not use up that much oxygen. And then other organisms would differ in their clearance rates. For example, functional group 6, their chaetognats have a lower clearance rate because the chaetognats just tend to hang around in the water column and wait to ambush their prey. So one thing you might notice is that the functional groups generally differ according to feeding strategies. So there are species which are current or filter feeders. There are species which are cruise or raptorial feeders. And then these could bridge between taxonomic groups. So like you have a current feeder that's a crustacean, and you have current feeders that are soft-bodied, and the same for raptorial feeders. So a trait-based perspective also, when you don't focus in the taxonomic composition of your community, you can focus on the functional composition of a community. And the same thing for ambush feeding strategies. There are ambush feeding soft-bodied zooplankton and ambush feeding crustacean zooplankton. And looking at, for example, feeding strategies is a good first step in using a trait-based approach. And that is important because there are trade-offs between behavioral strategies and other traits that affect organisms' morphology and physiology. We also know that feeding strategies reflect the different zooplankton functional groups and how these different groups target different size classes of prey. So we know that, for example, say euphausiids and ostracods target a specific phytoplankton size class, and you have your carnivorous raptorial zooplankton eating other zooplankton. So this is also useful so functional grouping and the trait-based perspective is also useful when we try to simplify how we organize, say, hundreds of species, and we try to figure out the marine food web. Right, now let's scale that up to the level of zooplankton communities. Here I will use the Northeast Pacific Ocean as a case study in applying trait-based approaches. In our work, so this is the Canadian Northeast Pacific Ocean, just to give you an idea. So this is at the other side of the Pacific from, say, the Philippines. At the north is Alaska, and south of this would be the US. So in this figure, I show the distribution of over 3,000 net samples that we use in our analysis. We analyze 2 decades of zooplankton vertical net samples from various monitoring programs. Right, and the colors of the points show when the samples were collected. So yeah, so we have samples from 1996 to 2014 and beyond. Okay, so what's important is that we can analyze like community data through a taxonomic point of view, right? So through a taxonomic analysis of the zooplankton of the Canadian Northeast Pacific Ocean, in this map, we looked at its biogeographic distribution. And here we have like a summary of how the different biogeographic zones from the offshore ocean to the deep shelf to the nearshore ocean and to estuarine fjords vary in terms of, say, zooplankton abundance, biomass, and community composition. And from using a taxonomic perspective, we can also start to identify the different environmental drivers affecting community composition. But there's still quite some limitations with like the insights that we get from a taxonomic analysis, even if it already tells us a lot about what makes up a community and what are the factors that affects it. Particularly, it's hard to really quantify what that really means in terms of looking at the ecosystem beyond just the zooplankton community. So we attempted to understand that by linking the taxonomic data set with the zooplankton trait database. So the trait data and the functional groups can also be applied to estimate community level functional composition and processes. So, and like, yeah, community level trait processes. So when we have the taxonomic data and count, so for example, you go out to the field, you tow your net, and then you do your microscopy, and you know the number of species within the net, you can match the trait values of those species from what you get from, say, the trait database, or you measure your own traits. And then using that, you kind of would erase the identity of the organisms, and then you would just like focus on the traits that make up the community. So for example, when we compare the different biogeographic zones of the offshore, deep shelf, and nearshore ocean, we can look at it through like taxonomic composition, but we can also look at it through the composition of functional groups. So the bar plots show, say, the relative contribution of the different functional groups to total community abundance and total community biomass. And what this demonstrates is that in terms of looking at the abundance, most of the zooplankton actually in the community are from functional groups 1A, which are your small crustaceans, and functional group 8, which are also your small brooding omnivorous crustaceans. And those like would span different taxonomic groups. But when you look at biomass, it gets more complicated. And so it has a different point of view, but wherein majority of the biomass could be accounted for by large crustacean current feeders, but then there's also a diversity of soft-bodied functional groups that contribute to biomass. So another way to look at this beyond just like functional grouping is when you combine zooplankton abundance and biomass data with species-specific trait values. So I'll just give an example of the many traits that you can look at. So one example are community level trait values for excretion rate. So here in these plots are the total excretion rate of ammonia and phosphate at the community level, and these are averages that are weight specific. So you just divide it by biomass, but that's not important. What's important to know is that looking at excretion rates is an important process in the organism's metabolism, but it's also an important process in terms of the ecosystem level because that affects the recycling of nutrients. Because the ammonia and phosphate that is excreted by zooplankton then become available to be used by phytoplankton for recycled primary production. So through this approach, you can actually quantify or have an estimate of how much say nitrate and phosphate and nutrients that like the zooplankton community would contribute to the available nutrients for your phytoplankton community. Another example is in terms of stoichiometry, wherein, for example, we can compare the carbon to nitrogen ratios between the different biogeographic zones, and we also look at the carbon to phosphorus ratio. We can look at water content. So we can get these community level estimates of the different, say, chemical properties or biochemical properties of the community, which tells us about the quality of the nutritional quality of zooplankton in that community. So you don't not only have an estimate of quantity, which is like biomass and abundance, you also have an estimate of nutritional quality, which is something you can get from, say, biochemical composition and water content. And this is important when you're looking at zooplankton, especially as prey for fishes, which have various nutrient requirements. Right, and I'm gonna skip, I have like one other example, which I'm gonna skip through. So here, this just shows that, when you have like a large trait database, you can look at the various different traits at the community level. So even if you just have, say, count data of zooplankton, you can use the trait database to, say, estimate the total carbon weight, carbon content of a community. You can get respiration rates, which leads you to zooplankton production rates or secondary production rates. And so that's very useful in modeling, and say we already shown ammonia and phosphate excretion rates, and you can also have estimates of feeding rates. That can also be used in understanding the food web. Right, and I'm gonna try to end with these couple of slides. So those examples are things that are happening at the regional scale. At the global scale, we have a good idea of the global distribution of zooplankton biomass. So here is one of the examples of aggregated data of net zooplankton biomass. So we can understand the global trends of where zooplankton biomass is high or lower. But understanding global trends using trait-based approaches is still limited. There are few there are few studies which have attempted this, but it has been focused on groups with the most trait data so far, such as copepods. Still, those have been very insightful in starting to estimate the global distribution of traits and looking at functional ecology at the macro scale. Traits are also very important in parameterizing models, and models designed with a trait-based approach in mind have been helpful in unveiling the underlying ecological mechanisms at the global scale. So for example, you have models, you have like food web models that can be designed to focus on traits such as body size and the predator-prey size ratio. And these have been very useful in improving our estimates of the importance of zooplankton in the marine food web, especially in highlighting and quantifying the roles of gelatinous and soft-bodied zooplanktons. They're often previously underrepresented in our global food web models. So I hope I gave you like a good survey of the many ways in bridging the individual level and community level processes in zooplankton ecology through a trait-based approach. I just like want to like list some next steps that I think are going to be very useful in advancing the field of trait-based zooplankton ecology. So one of the important future directions that you might be interested in pursuing is, for example, in terms of trait data, even if we have like zooplankton trait database, we still need to do a lot of data rescue and aggregation, especially for trait data that is like hidden in gray literature. So you might have a lot of information, say, in your lab journals or in your logbooks or in supplementary materials for your thesis or papers, even if it's just like, say, body size or carbon content for one species of copepod, like that is still one extra point in the big picture of zooplankton traits. So those are all useful. We also need new trait data measurements, especially for smaller zooplanktons. A lot of the studies measuring zooplankton traits are for zooplankton in higher latitudes, which tend to be larger. And so we need trait data for smaller zooplankton species in the tropics, but also earlier life stages, which are like really harder to handle. And there's still a lot of need for data management and accessibility to make, like, say, the zooplankton trait database more accessible and useful for people who want to use it. In terms of theory, we need to understand the trade-off between the different traits, and that would require a lot of experiments. So we can run experiments, simple experiments for, say, one or a couple of species, and we look at the interaction between, for example, body size, carbon content, and respiration rate. And these simple experiments are very useful, especially if we aggregate many of these in understanding the trade-offs between traits. It's also important to look at the mechanistic links between traits and ecosystems functions, especially for zooplankton. And also, there's also a lot of work that could be done in terms of applying trait data and trait-based approaches for zooplankton ecology. For example, using the trait database to get empirical estimates of community-level processes, as I've shown for various regions around the world. You can also improve model parameterization and structure to have a more trait-based approach or trait-based perspective rather than focusing on the taxonomic representation in models. Yeah, and a big challenge for the community is also to do trait-centered surveys, where instead, when you go out to sea and you cast your net, instead of like simply just instead of like counting different individuals and like doing taxonomic analysis, you can do your surveys where you actually just like measure, for example, overall body size, or you measure individual level elemental compositions and stuff like that. And you can use the wider library of zooplankton traits to make more insights out of those trait-centered surveys. And then finally, there's a huge need for measuring community level processes, especially for zooplankton. So instead of just like capturing like zooplankton through nets and then counting them, it's also important to say do mesocosm experiments or doing like live rearing of zooplankton communities and seeing, say, overall respiration rates and feeding rates. And all these would like eventually scale up like the different, whether like all these small experiments will scale up to like a huge global data set and database of zooplankton traits, and which can be applied to other research. All right, so that is it. Thank you for your attention. If you are interested in using the zooplankton trait database, or you're trying to find ways on how to analyze your, for example, community composition data and convert that to estimates of ecosystem functions, you can access some of my code in my GitHub account. I want to thank my many collaborators that have contributed to this study, and also the funding agencies that have funded my research, and especially to Louis and Christy for the invitation to do this talk. All right. Thank you.
Thank you so much, Dr. Patrick, for a very interesting and comprehensive talk. It was very exciting to see a lot of studies that you have presented and cited. And also, as someone who works also in zooplankton, it's very satisfying to see how trait-based approaches at the individual level can be translated to a better understanding of community processes of zooplankton, and eventually on a larger picture of in the ecology and in the marine ecology. So I'm sure everyone has a lot of questions for our speaker today. So as our moderator for the open forum, may I call in Dr. Mark Louis Lopez.
Hello, everyone. Thank you, Patrick. That's a really awesome presentation, especially the figures. It's really nice and really
Thank you.
effective and like conveying the message. Yeah, so everyone, if you have questions, feel free to raise your hands or feel free to send your question to our chat box and I'll be reading it. So we have two people who's raising their hand, and I'll try to unmute Henriel. Okay, let's go for Charles. So you can unmute yourself, Charles, if you have questions. Charles? Okay, so maybe while waiting, we have consolidated some questions. I think the first one is like most of this audience are undergrad and grad students who are currently doing zooplankton ecology work in the Philippines. Uh, can you give us like an overview? How did you start with a functional based or trait based analysis? Like how this all started? Because like we already have the capacity of collecting zooplankton communities and doing the basic and the taxonomic work for community structure assembly. So how can we transition to trait-based approach?
Yeah, I think like the main reason why I got into this specific field and approach is that I started with trying to, understand community level. Like I tried to understand zooplankton communities through a taxonomic perspective, and that's really challenging, especially when you're trying to deal with like a diversity of organisms such as zooplankton. So you have like you have copepods who are doing their own thing. But still you have to consider pedognaths that could be their predators. And then you have to consider jellyfishes. And then there's a variety of different gelatinous organisms and like stuff like that. And then, but then we often aggregate them in terms of all the whatever species we get in a plankton net. So it gets really challenging of say, when when you get data sets like community composition, and then you try to answer a question of say what is like the part, what portion of this community is actually providing nourishment to fish, or like what is a portion of this community that is actually contributing to say carbon flux. So it's hard to like answer those questions when you just like look at it through the point of view of taxonomy. It can be useful if you know a lot of a few species. For example, there are very well-studied species, say in the Antarctic. Like you can like study a lot of processes of the Antarctic based on the biology of, say, Euphausia superba. But in like more diverse communities, like it's really tricky to point those out. And so I tried to look for a strategy in gaining more insight, making the most out of like our zooplankton net data, because it takes so much effort to do taxonomy and to sample zooplankton. It has so much effort to even get to the point where you have like a community composition count. And if you just like struggle with interpreting it qualitatively, you need like another strategy to make the most out of those trait data. And so what I do is just like, given these taxonomic counts, I have like a list of species or genera, and I just like query the trait database and see what are the estimates for those. And then it's basically just matching your species table with the trait table. And if you have like an idea of abundance and biomass, and you have individual level traits, for example, carbon content for one copepod species, or say respiration rate per individual, you can just multiply that with abundance and you get the population level or community level estimates of those rates. So yeah, that's like one of the strategies when you can on how like you can apply this approach.
Sounds good. Thanks, Patrick. We have few questions in the chat box right now, but like while we are on the topic of like the database and how to use it, there's one question on we are studying zooplankton communities in the Philippines. Can we use the database that you created, considering that like are there database for tropical species, or is it limited to the Northeast Pacific region?
No, the trait database has a global range, a global coverage. Majority of it would be species from other zones of the ocean, but there will still be a lot of tropical species and also smaller species. But the important thing when you think of like using trait data is that you can also start to not think about the biogeographic distribution of species. So if you're working with like species that are not in the trait database, but they're of similar sizes or similar, say, feeding strategies with species that have trait data, you can try to estimate, the characteristics for the species that you have from species from other parts of the world. So there are strategies in doing that, especially when you like stop thinking about things in the local or regional perspective and just try to make use of data from the global trait database. But the challenge of that is that smaller species, like very small zooplankton, often have fewer trait values. So it depends on the size range of what you're working with, if there's actually even available information for that size, size of like zooplankton. So yeah.
It's a really good perspective. Uh, I have a lot of questions, like personal questions, but I'll go with the questions from the audience. First question is like, can we run functional traits and group based only on major morpho species group?
Yeah, that's like a great question, and also that's quite a good approach. I don't know if I still can share my screen. Can I do that?
Yep. I think you should be able to do it.
So, for example, like when you look at functional grouping, you can observe, like here, that they're primarily differentiated, say, by size. So you have like smaller individuals, larger individuals. They're also, morphology tells you a lot of things. So like species that have high water content often are functionally very similar. Species that have, say, specific morphological properties such as filter feeding apparatus are functionally similar. So you can definitely work with a trait-based approach without even knowing species level stuff. And this is very applicable to when, for example, you're working with imaging data, right? So when you have imaging data, you can identify families or major groups, but you can what's useful is to actually get morphology, and from morphology you can estimate so many other traits just in terms of body size and water content alone. So yeah, so that's like a great strategy.
Thank you, Patrick. Next question is like, did you also study diel vertical migration of zooplankton? Since DVM is a plastic trait, are there any updates to improve the analysis for this trait?
Yeah, that is a extremely good question, and one of the most, I think, challenging traits to work with. I not personally work on that, and there are a few papers that work on DVM and traits. The tricky thing with DVM is that it's not just like species specific, it's region specific. The rate of vertical migration and the rate of I mean, the distance of these migrations and when they happen is like very different, even for the same species, very different between regions. But the zooplankton trait database has the records of some of the observations of vertical migration. But yeah, so like that's a bit limited, but just like consider how, you like when you study DVM, like you can there are kind of like two ways to do it. You can tow nets at different zones of the ocean, and then you try to find out where species are found. But you can also use acoustic sensors or imaging sensors that would not have like a good estimate of taxonomy, but will actually have like a good estimates of vertical migration. So yeah, so there are ways around it, especially when you try to look at the community through different points of view.
Sounds good. Yeah, we're a bit over time, but like we still have 3 more questions in the chat box. Let's go quickly. What are some challenges in developing a comprehensive matrix of key traits for diverse zooplankton groups? I think this would be very applicable like in tropical region. So like, what are the common challenges that you've encountered?
Yeah, I think there are a lot of the challenges would be related to how much accuracy do you want to have? Because if you want to be very accurate, if you want every single species to have a very specific trait value, then it might be a challenge to populate that matrix. But if you kind of understand that, similar species would have similar morphologies and sizes, and they have like likely similar traits, you can get away with working at it at like a broader or like more coarse taxonomic range, and that will really help you with dealing with those challenges. So I think my recommendation is that first look at it like at the most useful coarse functional scale that you can, say major taxonomic groups or like families, and see if you can get insight from that. Because if you can get insight from that, then maybe that's like a good enough level. But if not, you can go down to another level. So like try to not approach it right away from the point of view of individual species, because like a lot of these organisms are more similar than we think, even if they're genetically different.
And at some point, like you need to weigh like the scope and at the same time accuracy, especially
Yeah,
when you're dealing with like regional scale or like global scale.
yeah.
They keep on coming in, but like I think I'll narrow it down like to 2 more questions. Is it possible to do the trait-based approach clustered on body forms or life forms than off species level in a horizontal distribution? I'm not sure if
Hmm.
that's like
If I'm understanding, if you only
Mhm.
have like body form, life form
Yeah,
data.
body form and life form data of species level in a horizontal distribution.
Yeah, I mean, if you have like species level information for those, I think, yeah, well, one thing like you can do with a trait-based approach, especially now that's it's in like a framework of how the different traits related to each other. If you have information of say body form, life form, or size, you can use that to estimate what the other trait values might be, and you just like invoke different theoretical stuff. Like for example, you know, one example would be like for I don't know, like for gelatinous organisms like jellyfish that are in a more like for example, jellyfish that have longer tentacles tend to be more ambush feeding, and then jellyfish that have like are are flatter and have like shorter tentacles, have like a pulsating motion, tend to be cruise feeding. So there are ways around it. You just need to explore the trade-offs between the different traits and see what could be useful for you. Yeah.
Sounds good. Yeah, we still have a lot of questions coming in, but like you provided your contact information, so I think
Yes.
if they have like specific questions, they can reach out to you and then like discuss it in more details. But like at this point, I just want to maximize like the opportunity and the platform because like I know that the database is a really good contribution because it's like one of the limiting factor in doing functional or trait-based approach, even like
Yeah.
in freshwater ecosystem. So if some of our audience are like they want to do some data rescue because they have long-term data for some zooplankton communities in the Philippines or within Southeast Asia, is it possible for them to contribute data set to the database and how they can do that?
Yes, and it is completely possible, and I really encourage that. If you want to organize like a data rescue like project, and if you need help, I'll be very happy to like give recommendations on how to do this like strategies in doing it. Specifically for the trait database itself, we mostly use trait data that is already published or has gone through peer review. Fortunately, now there are like publications where you can peer review data sets. You don't need to like interpret them. You can aggregate trait data. You can do a data rescue project and just present that database, put that through peer review, and that would count as a publication. And then from that point, it can be used by like larger databases because often that like biodiversity and trait databases would require publication. But given that there are a lot of these journals that have like data specific, say, subjournals or versions of papers, and then that is not a hindrance. So like what is really the thing that we really need is like for people to do the actually challenging work of just like going through all these like logbooks and a lot of old data sets and just like digitizing them. But if anyone's interested, please feel free to reach out and I can like help out and just pointing you to different ways and how to best make use of your time. So yeah.
Yeah, that's amazing. Like if there's an undergrad or like graduate students looking for a thesis project, so
Yeah.
you
And
know the
and
way.
honestly, that would be very like especially for understudied species. Like even if you do it for like 1 to 10 species and you like measure literally like a couple of traits, that's like a really huge contribution, especially when you look at how sparse the coverage of trait information is. So yeah.
Yep. Thank you so much, Patrick, for this presentation. And yeah, so
Thank you.
you provided your contact information. So if there are some audience who want to connect with Dr. Pata, feel free to reach out. And I think that's the end of the open forum. So we're a bit like 10 minutes over time. So I'll pass it over to Darlene to close the program.
Thank you, Sir Louis and Dr. Patrick. So before we close the program, we would like to first award the certificate of appreciation to Dr. Patrick. The citation says the certificate is awarded to Dr. Patrick Tata in recognition of his outstanding contribution as keynote speaker of the presentation titled Bridging Species Level and Community Level Processes Using Trait-Based Approaches in Zooplankton Ecology at the fourth lecture series on Philippine Planktology on August 7, 2024. Signed, Dr. Christy S. Acabado, Director of the Institute of Marine Fisheries and Oceanology of UP Visayas and Vice President of the Plankton Society of the Philippines, and signed also by Dr. Mark Louis Lopez, the current administrator of Philippine Planktology. So once again, we are very thankful for the investment of your time and effort, Dr. Patrick, for today's lecture. We will send you the copy of the certificate via email as well.
All right, thank you. And then thank you everyone for sticking around, and thanks for organizing.
Thank you, sir. So before I head on to the closing of this program, I would like first to highlight these degree programs offered by the Institute of Marine Fisheries and Oceanology at the UPVC course. So we have a Bachelor of Science in Fisheries and also Master of Science in Fisheries in Major in Fisheries Biology and Master of Science in Ocean Science as well as PhD in Fisheries. So for more updates about upcoming events in both the Institute of Marine Fisheries and Oceanology and Philippine Planktology, as well as a Planktonish Laboratory, you can follow these Facebook pages you can see on your screen. And our last lecture would be on August 12, 2024 by Dr. Leching Chu, and she will discuss about theropods and what they tell us about the evolution in the open ocean. So we look forward to seeing you all in the next lecture on August 12. And thank you so much, everyone, for attending today's event. You can the Q&A box to receive your e-certificates. Thank you so much.
Thank you, Patrick. Thank you for your time.
Thank you, Patrick. We will be sending you email soon.
All right, all good. Thank you. Sorry for
Yeah.
going
Well,
over
Thank
time,
you.
but
oh, no,
Oh,
no,
it's
we're
perfect. It's
good.
okay.
Yeah,
Yeah.
really great
um,
presentation.